All right, thank you very much. So yeah, I'm a PhD student here at Murdoch in my second year, and we're from the Vector and Waterborne Pathogen Research Group, and we're interested broadly in vector-borne pathogens and waterborne pathogens and their epidemiology and monitoring. So what does that mean essentially? So we have samples from a range of different sources. I myself work on ticks, so those little arachnids that will bite you when you're out bushwalking. We have other people who work on waterborne pathogens and parasites, and also parasites that can infect you through the fecal-oral route, and that's of course intrinsically related to water as well. And essentially we use a range of molecular tools, so DNA-based tools, to work out what bacterial or protozoal or parasitic organisms are in these samples, and then what implications that has for epidemiology, monitoring, disease control, things like that. So our samples can range anywhere from fecal samples from a herd of cattle that have a parasitic infection, and we want to know some dynamics maybe about the population of parasites, maybe their intra and interspecific population structure. We might have for example, water from swimming pools from around Australia, and we're interested in what's being shed into those pools from people and then potentially reinfecting the population. Or we might have arthropods, which we know are great vectors of many diseases, and we're interested in the dynamics of those infections or the dynamics of the transmission of what goes on there. And to do all of these things, we use one core toolkit, which is metagenomics. And what metagenomics is essentially bulk extracting DNA from these samples and sequencing that DNA. And we can sequence it, I'll explain a little later, either in a targeted manner or in just a sort of bulk sequence everything sort of manner. And then through bioinformatics, which is where Pawsey comes in, figure out what bacteria or parasites are where in the sample, how abundant they are, and various things like that. So it's one core toolkit that we can use to answer many, many questions about the biology of these organisms. And to do that, we use benchtop sequencers, all of which are available here at the uni. So in the last 15 years or so, DNA sequencing has grown exponentially in the amount of data that it can output for researchers. Traditional sequencing is Sanger sequencing that you see there on the left, where you input one DNA sequence and you get one DNA sequence out. There were then advances in pyrosequencing, where you could generate maybe 1 million sequences, normally around 100,000, but up to 1 million. And now we're in the Illumina era, where we're routinely, in a single run, generating 25 million sequences. And we also have the capabilities here at Murdoch to generate up to 600 million sequences per run. How does that equate into gigabytes of data? From an Illumina MySeq run, which I guess would be our minimum throughput that we have going at the moment, it's probably around 15 to 20 gigabytes of data per run. And when we get up to the Illumina NextSeq, which is the big daddy we have on campus, that's generating up to 250 gigabytes of data per run for us. So it's a lot of data to be able to process at once. And certainly a laptop like this, which used to do the work for us, is no longer really relevant in this game. So I mentioned briefly before, there's a couple of different types of sequencing we do. The one on the left is called Amplicon metabarcoding. So in this sequencing, we're generating one gene barcode from every organism in a sample. And this barcode then allows us to identify what that organism is and how abundant it is in the sample. So this workflow is really great for identifying the diversity of organisms, for discovering potentially new and novel organisms. But to be honest, a laptop can still process this sort of data. So this would be our sort of 15 million sequences from a MySeq run, would be the Amplicon metabarcoding side of things. When we jump over to whole genome sequencing though, we're generating a lot more data, and it's a lot more computationally expensive to untangle that data. So when we're doing whole genome sequencing, or especially metagenomic whole genome sequencing, we have a particular organism or a particular group of organisms from a sample. Maybe it's a tick sample that has the tick DNA in it, it has the host blood DNA in it, it then also has microorganisms in the tick's gut, and then it might have a small amount of a pathogen DNA that you're interested in as well. or it might be a single source sample, like just water that you know is infected with one organism, and you want to sequence the genome of that whole organism. And the way that this sequencing works is essentially the whole genome of this organism is chopped up into lots of small fragments, maybe 200 or 300 base pairs long. We then sequence those small fragments, and then later we have to reconnect it all back together into a contiguous genome sequence. And that's where the computational resources really come in. It's really at this third stage in our pipeline that we started doing whole genome sequencing, and we started running into huge problems with just not being able to process our data in a timely or efficient manner. And that's, of course, where Pawsey came in. We faced three problems, essentially. We needed more CPUs, and really we needed more RAM to be able to do the sort of assemblies that we wanted to do on the whole genome data. We needed somewhere to store this data while the analysis was happening. So we had, 4 or 5 hard drives per person in our lab, terabyte hard drives, clogging up desks, being transferred between computers, and it just wasn't efficient at all. And the third problem we faced is that our data wasn't yet large enough or high throughput enough to really justify the access to Magnus or something like that. We did have Magnus access, which we used, underused probably. And the reason for that is our workflow isn't high throughput in the sense of we're not doing a sequencing run every day and feeding that data into Magnus. We have one run, we analyze the data, it's done, and then we move on. And so those sort of, that computational style of doing things wasn't really applicable for Magnus. And the solution was Nimbus Cloud Services, which has been fantastic for us. So essentially what cloud services are is connecting through a Unix environment to virtual machines or CPUs at the Pawsey Centre, and having access to store your data there, do your computational analyses there, and then transferring results back to you. But the other thing that was really invaluable was the training that we got from Pawsey. So I think it's suffice to say everyone in our lab sort of had a very basic understanding of Unix and command line, bash, and that was it. And obviously to connect to these sort of resources, we need more understanding of how to communicate with the VMs, what can we do on the VMs, what can't we do on the VMs, how to be secure on the VMs, all of the sort of things that we listed here was sort of a great experience that we had with Pawsey, upskilling our entire lab to be able to use these resources. And then also the day-to-day things. How do I use the Unix environment? Why isn't my program working? And what can I do here with things that were really invaluable, that were, a really nice interaction between us and Pawsey that really upskilled our entire lab, including our lab heads, which was good. So what have we done on Nimbus? Our lab has 2 Nimbus VMs at the moment, a smaller one, which is this one I'll talk about now, and then a slightly larger one for genome assembly. One of the VMs that we have at the moment is a dedicated BLAST server. So BLAST is the basic local alignment search tool, which is essentially a way of comparing your DNA sequences to a reference database of sequences. And the reference database we use most often is called NCBI's GenBank, which is a database that contains genome sequences of every sequenced organism in the world. It is a massive database. I think it's upwards of 200, 300 million sequences in there from different organisms. And obviously comparing your data to that database is computationally resource expensive because of the way the algorithm is designed. It's not necessarily designed initially to be able to parallelize very well. So that's an intrinsic problem with BLAST. And so we just needed more resources to do it. So we've got a dedicated VM now to set up that, which has increased our blast times 15-fold from doing it either a remote blast to a server overseas somewhere or doing it locally on our computer. 15 times, which is pretty amazing. And that's something core that we use basically every single day. Now, there are other resources that can do this sort of thing for you. This is nothing special that we have set up that is unique to Murdoch or unique to our group. I think every researcher who deals with DNA or protein sequences uses this tool. The fantastic thing about having a Nimbus resources dedicated is that we are the sole user of that resource and we can run it 24/7, 7 days a week. And I think that in itself is intrinsically one of the reasons why our productivity in terms of blast searches has gone up so much, because we don't have to queue, we don't have to share CPUs with other users. And the other resource we've set up, this is my VM. I've set up a VM which I've called my Bacterial de Novo Genome Assembler, or my bad gas. So essentially this is a 16-core VM with 50 gigs of RAM and a terabyte of storage, which I've pre-installed pipelines on which have all the tools you need to go from a raw DNA sequence or raw DNA sequences to a fully assembled bacterial genome or in genome contigs. It depends on how good your data quality is. And this is achieved, as opposed to maybe weeks, can achieve this in a 24 or 48 hour period if you have the right sample. With metagenomic samples that I'll talk about in a minute, it's a bit more timely. But at the end of the day, without this resource, we wouldn't be able to do the type of work that I'll show you in a second. So the type of work that I've been doing on these VMs is assembling bacterial genomes from a metagenomic sample. And one of the organisms I've been working on is this guy here called Borrelia tachyglossi. So Borrelia is a genus of pathogenic bacteria that is spread or transmitted by ticks around the world. So in the northern hemisphere, it causes a disease called Lyme disease, and throughout Africa and Asia and even into Europe and North America, it causes another disease called relapsing fever or tick-borne relapsing fever. And just 18 months ago, our lab identified the first ever Borrelia species here in Australia. And we are, of course, incredibly interested in this bacteria and whether it might be causing disease in peoples or animals and just about its general ecology and life cycle. So we identified this in echidna ticks. The problem was we had a tick, and as I said before, it was a metagenomic sample, so we had a tick full of tick genomic DNA, echidna blood genomic DNA, as well as all the microorganisms associated with the gut and the salivary glands of the tick and the skin of the echidna and the blood of the echidna, and then also a very small amount of this pathogen DNA that we were interested in. So we bulk sequenced this sample on the NextSeq, which is the big daddy we have here on campus in the SABC sequencing facility, and we generated 1,266,345,104 sequences, which is about 260 gigabytes of raw data. I then fed this into our Nimbus pipeline. Remembering, of course, we've got this huge mix of DNA that we need to untangle. So at the end of it, the Borrelia DNA that I was interested in assembling was less than 1% of the total DNA in that sample. And this is where the other VM came in really handy, that BLAST VM, because BLAST is a really great way to be able to untangle these mixtures. You have the ability to take your sequences and compare them to every known Borrelia genome and just pull out those hits that you wanted. And the ability to run that 24/7 on the dedicated BLAST VM meant that job was done in 3 days as opposed to a week, 2 weeks, 3 weeks maybe on another resource. So as I said before, the fact that we were the sole user of this resource made it incredibly possible. And now I have the entire genome of this organism sequenced. Why is that a big deal in itself? The genome, you've got to remember, tells you everything you need to know about that organism. So we're going to use this genome to identify genes and gene functions, which means that we can characterize metabolomic pathways, as in asking, how does this bacteria feed itself? And that's incredibly important when it comes to infection and transmission dynamics. We're also using this data to scan through the genome to look for virulence factors and pathogen islands. So genes which might increase its ability to invade human cells, for example, or genes which might increase its resistance to antibiotics. And again, that relates to pathogen mechanisms. And the other thing we're going to use this genome for is to identify antigenic proteins. So looking into the future, asking how do we test whether people are infected with this? The most common way to do that is with an antigen test. And so the basic of developing those is you need the DNA sequence for the protein, and then we can move forward and develop those tests. And all of these things would not be possible if I couldn't assemble the genome. And I couldn't assemble the genome without having the resources and the training that Pawsey supplied, which was really invaluable. Just summing that up, the fact that the support and training and resources together that were supplied by Pawsey were really integral to my research. I'm not a computational biologist. I'm a biologist. I spend 99% of my time in the wet lab with a pipette in my hand. And without having this, knowledge given to me by Pawsey and the resources, this sort of project would have been completely unfeasible to myself or my supervisors. So, thanks.